I'm Evelien Adriaenssens, or Evelien if you speak Dutch or Flemish, and I'm a group leader at the Quadrum Institute Bioscience, and I'm also the Vice President of the International Committee on Taxonomy of Viruses. Before I was the Vice President, I was the Chair of the Bacterial Viruses Subcommittee of the ICTV, meaning I was responsible for all of bacteriophage taxonomy. We've had some recent changes to bacteriophage taxonomy. We have two major changes. One of them is that we have changed the species naming conventions from a sort of free form where the species name could be bacteriophage T4 or Escherichia-phage T4, something like that, to a binomial system where you use the genus name first and then have a specific species indicator or species epithet. So we've had to change thousands of species names like that. And the other major change that we've had is that we went from a morphology-based system to separate bacteriophages into families. We went to a genome-based system so that we can use sequence information to do taxonomy. We've had some challenges implementing these changes. For the naming system, it was coming up with new names, and as well we had to convince people that this was a good thing to do, and we've had to then put in proposals to change thousands of names, which was not easy. And for the taxonomy, for the changes in the morphology-based taxonomy, we had to do large-scale data analysis with thousands of sequences to show that the changes that we were proposing were actually needed and would improve taxonomy. For me, the impact of the new naming system to the scientific community is really that we want to make the distinction between a bacteriophage or a virus and the species it belongs to. So if you think about the hierarchical taxonomy, where we currently go from realm all the way down to family and subfamily, genus and species. The species represents a unit of evolutionary history, and it's currently the lowest that we define. But each species has multiple viruses or multiple bacteriophages associated with it. So to make that distinction about this one virus or this one bacteriophage that you work with in the lab and the species it belongs to, it's important to have two different names. And we didn't always have that in the past. So I think this will be very useful to make that distinction for scientists. We've received mixed feedback on the system, the new naming system. So it depends on what area of virology people belong to. We've had good feedback or we've had bad feedback. Some people were very attached to the names of their virus species. Even though the name of the virus itself did not change, the species it assigned to changed. And this was kind of difficult for some people to accept, as well as trying to come up with the new names and people discussing what the proper name would be. You know, can it be a name of a person? Would that be appropriate? Is it better to have, you know, a name that is associated with a place or something that is totally agnostic of these type of personal connections that people might have? So overall, I think the community has largely accepted the new naming scheme, but it's a work in progress. So the naming system itself, I don't think we're planning any big changes to it because it's been a shock to some scientists to get this. Overall, we've had to change thousands of species names. So we hope that this can kind of settle for a while. The morphology to genomics-based taxonomy changes that we've done for bacteriophages specifically, this is something that we're expanding to make sure that we can incorporate data from sequence-based only studies so that we can use metagenomic, viromic, metatranscriptomic studies to inform our new taxonomy and to get new species, genera, families created based on sequence diversity and virus diversity that we find in nature, but we're not maybe being able to culture them or visualize them under an electron microscope. So the ISCTV, there are some emerging challenges that we are trying to address, and the main one for me is dealing with the influx of sequence data. So we're in the era of big data. There's so many sequence data sets out there, so many, especially metagenomic or sequence data sets, metaviromic, viromic, metatranscriptomic, however you want to call them, where we identify viruses only based on a contig in a bigger data set. And dealing with the challenges that comes with that in terms of quality control. Is the genome complete? Is this virus observed elsewhere? Is this truly a virus, or is this, an artifact of assembly? These type of things. These are the main challenges, and we are en route to addressing them. But it will take some time, and it will take the effort of a lot of people and a lot of volunteers. So the ISCTV is different than other taxonomies because we deal with nomenclature and classification at the same time. So we both build the framework using certain demarcation criteria to build the kind of evolutionary history and tree that then guides our taxonomic classification, and then we are giving them, assigning the names. And we do this by committee. This is why we're the taxonomy committee. So that means that people present proposals, and we then assess them and vote on them in multiple steps, so that everybody who wants to can have their say, and it's not necessarily the person who publishes something first which doesn't match with something somebody else has published before that. So we kind of take all of these things into account to come up with a framework that hopefully works for everybody and is universally applicable. So I think that kind of helps the ICTV help the global scientific community. We've had a lot of major changes in the past. So currently we're largely focused on making sure that these changes are implemented well and that the community is really up to speed with what we've done. So we've focused a lot of our time recently on getting communities of experts together and discussing these changes and how we can go forward with them. There's no immediate plan for another big overhaul of virus taxonomy or bacteriophage taxonomy. I think we've shocked the community enough in the last couple of years, and it's now about framework is in place, and it's about future stability and implementation of what we've done. So I'm currently the vice president, and as part of that, my role is to be the editor-in-chief of the ICTV report. And so the ICTV report is a set of online book chapters and profiles in the Journal of General Virology that describe families of viruses. And it's very rewarding to do this at the moment because I get exposed to lots of different types of viruses. So I try to make sure that I read and sign off on every chapter that comes across my desk. So I read about archaeal viruses and human infecting viruses, eukaryotic viruses, on top of all of the bacteriophage work. That's really my area of specialty. So getting to know the whole of the virus sphere, as I call it, is very rewarding to me. The advice that I would give young virologists is to not be overwhelmed by all of the information that is out there. We've been working on this for so many decades, and we're really standing on the shoulders of giants when it comes to people who have started taxonomy. Do your reading. Go to our website, ictv.global, see what taxonomy proposals have been submitted, see what the current structure of the taxonomy is, see what, depending on what part of the virus sphere you're in, if you're working with RNA viruses, evolutionary history is different, so the rules in making the taxonomies are different than if you're working with bacteriophage, double-stranded DNA bacteriophages. So recognize that there's no single way to do taxonomy for all viruses at the same time. So you might need to specialize in a certain group of viruses, you might need to specialize in a certain set of tools that work for the group of viruses that you work with. And then don't forget about the other viruses, because sometimes people can get stuck in their own narrow way of thinking, oh, this is true for my set of bacteriophages, my set of viruses, so therefore it must be true for everything, and that is definitely not the case. So yeah, narrowing it down to don't be overwhelmed, do your reading, and think about the whole of the virus sphere.